Hi everyone. So we also need to make up this lab material. Now for next week's lab quiz, first of all, it will not be on Tuesday. Tuesday is your test day, so we'll move back the lab quiz to Thursday. So you will be responsible for the entire lab, and I'm kind of catching you up by making this video lecture. There'll be multiple pause breaks for you to kind of work through the questions before listening and working through the answers. So keep your eye out for those. Now, for the most part, I'm pretty sure I went with every group and double checked your answers about the cellular organelles. But if you're not sure about any of your answers or you'd like to double check with me, send me an email, but I'm pretty sure I went through that with all of you. Most everyone looked at the carmine particles and I answered most of your questions. I'm going to go ahead and go through those answers anyway, but I know everyone at least got to see the particles. So I'm currently on page 62, and this is where you made a slide that had water and you added these red particles that aren't living. Now, the first question, question number 3, asks what happened, and all of you guys saw movement. However, looking at question 4, these particles aren't alive. These carmine particles aren't living cells, and they can't move on their own. So the question is what's happening? Well, think about when you put that toothpick down, you had a very high concentration of these particles. Well, these carmine particles are then going to move from that high concentration to a low concentration. Now, if you look at that next statement on the bottom of page 62, it says that molecules of liquid are always in motion. This means water molecules everywhere are constantly moving and bumping into each other. As they move, they also collide with anything floating in the water, such as the carmine particles. So that's what's actually causing this diffusion, going from a high concentration to a low. It's these really small water molecules that we can't see looking at a microscope that are moving these particles around. So the question on the next page, page 63, says if we warmed up the carmine slide a little bit, the rate of molecular motion would be what? And so the answer to this is that it would be faster. So think about really hot water, water that's about to boil. Well, boiling water is moving quite a lot faster than, say, really cold water. So those molecules are going to be moving faster if it's warmer, so they're going to push those carmine particles a lot faster. Alright, so I'm going to go ahead and move on to activity 2, which is on page 64. However, I do want to note, be sure to read through these different paragraphs and notes. A lot of it is just a review of what we already went through, but it may help just to remind yourself what we're talking about. So these next few activities, activity 2 through 5, we did not get to do in lab. So make sure that you're following along in your lab book and answering the questions before listening to what I say. So what you're going to be doing in activity 2 is taking a dialysis bag, which I have shown here. A dialysis bag is a semi-permeable membrane. It's not a plastic bag where nothing can get through. Some things can get through, and this experiment is testing that. What can get through this semi-permeable membrane? So in this activity, what we were going to do was fill up this bag with a solution. In this solution, it has salt, it has glucose, and it has starch. And so we filled up this bag with that solution and we put it in this cup of water. Now in this water is absolutely nothing. There's no salt, sugar, and sugar is in starch or glucose. So absolutely nothing in this water. Those 3 molecules are only in the dialysis bag. So we're going to let it sit here for about 20 minutes, and at the end of this, we're going to test, did any of those particles move outside of the bag? So did any of the particles that were in here move into the surrounding water? So go ahead and look at number 5 on page 65, and it asks you what the initial weight is. And so this is one from my other class, and this weighed 20.2 grams. You can see that here. So go ahead and write that in that blank. Now go ahead and read through. Question number 7 or step number 7 asks you to write a hypothesis about how do you think that weight may possibly change and why. Go ahead and also flip to the next page, to page 66, and complete those comprehension check questions. Once you finish those questions, go ahead and restart this video so we can go over them and move on to activity 3. So go ahead and pause and complete those questions now. Alright, so I'm going to jump to the comprehension check questions. I'm not going to go over your hypothesis because your hypothesis is your own. So the first question asks, when someone uses a 10% glucose solution, what is actually in this solution? So it already tells you there's 10% glucose. So you know that would be your first blank, is 10%. Now the solute, which is glucose, and the solvent, which is water, is always going to equal 100%. So if we know 10% is glucose, then 90% would be water. The next question asks about the concentration of everything. So the first one asks, is the highest concentration of salt inside or outside the bag? And this would be inside. We put salt in the solution in the bag, but outside the bag we don't have anything. The highest concentration of sugar is also inside the bag. The highest concentration of starch is also inside the bag. However, the highest concentration of water is outside the bag. In this water, we don't have anything else. This is 100% water. Whereas inside the bag, it's less than that. I don't know the exact percentage, but it could be something about 70%. We definitely know it's not 100% though. And then the final part, if my dialysis tubing gains weight during the experiment, you'll know that water entered the bag. That's the only way it could gain weight. Nothing else could be entering the bag. If my dialysis tubing bag lost weight during the experience, you know that the water would have left the bag. It has to be left. That's the only way to lose weight in this case. Alright, so let's go ahead, now that we've developed our hypotheses and kind of taken a guess at what may happen, this is going to sit for 20 minutes. So while that's sitting, we're going to go ahead and move on to activity 3. So this is on the next page, on page 67. So we need to test that surrounding water. So we have the dialysis bag and it's sitting in water. At the end of that experiment, we want to measure, is there any starch, any glucose, or any salt in that water? If there is, that means, okay, that left the dialysis bag because it wasn't in there to begin with. Now we've actually already done tests for 2 of these. We've done a starch test. Remember, we used iodine. We also know how to test for glucose and other simple sugars. We do that using the Benedict test. However, we haven't done a salt test yet. So the silver nitrate test is a way to test for chloride ions. Remember that salt is sodium chloride. So this silver nitrate test, you can read a little bit more about it on the top of page 67, will test for chloride, iodine tests for starch, and Benedict's tests for sugar. Now in this activity, all you're doing is doing positive controls. You get out 3 test tubes, you put in that solution that you put in the dialysis bag. So that solution again had starch, glucose, and salt. And then we're going to test each of those. So 1 test tube for the iodine test, 1 test tube for the Benedict's test, and 1 test tube for the silver nitrate test. So here's what our results are. So before I interpret these results, go ahead and read through activity 3 just to visualize how we did these tests, and then answer the question on page 69, so number 11, and fill in the first 3 rows of the table on page 69. Once you've completed that, go ahead and restart this video and we'll go over it. Okay. So on page 69, question 11, it says if starch leaves the dialysis tubing bag, I expect the iodine test results on the water in the jar will be positive. because that means starch left the dialysis bag and entered the water. And so we should see that using the iodine test. If sugar doesn't leave the dialysis tubing, I expect that the Benedict's test results on the water in the jar will be negative. That's saying sugar stays in the tubing the entire time. It never enters that water, so we would expect that Benedict's test to come out negative. And then finally, if salt leaves the dialysis tubing bag, I expect the silver nitrate test results in the water will be positive. Again, salt was in the bag and it's leaving the bag into the water. Now this table is just a way to keep track of your results. These first 3 rows are for the controls. So the silver nitrate test will test the presence of chloride ions, so we use that for salt. Initially that color is clear, and you would have gotten that by reading the description of the tests. And the final color for us was cloudy, and so this indicates a positive result. The iodine test tests for starch. Originally this is yellow. Ours came out black, which means it's positive. And then finally glucose is tested using the Benedict's test, so here in the middle. Initially, this solution is a turquoise light blue color, and it came out a yellowish orange color. So that means that it was positive. So now that you know how to run these tests, we can now test that water left by the dialysis bag. So let's say this dialysis bag has now been sitting in the water for 20 minutes. So anything that was going to move, let's say, has moved. So if starch or glucose or salt was going to move, it's moved at this point. Now the first thing that we look at in activity 4, which is on page 70, is it asks what was the final weight. So one of the groups found that the final weight was 23.6 grams. And I also reposted the initial weight of 20.2 grams. So knowing this bit of information, go ahead and answer questions 2 through 4 on page 70 and start when you're ready. Okay, so if we look at question 2, our final weight again was 23.6 grams. And that means that the bag gained weight. We were at 20 grams and now we're at 23. So depending on what your hypothesis was, this could be wrong or right. Next, what you need to do is calculate the percent change in weight. So to do this, we take the final weight, so 23.6. We're going to subtract the initial weight, so that's 20.2, and divide by the initial weight, so 20.2. This is an equation you do not need to know for the lab quiz. I'm going to take out my calculator, do 23.6 minus 20.2, divide that by 20.2, and then you times this by 100 to get the percentage, so I get 16.8. So this is saying that the bag increased in volume by 16.8%. Now the final question, question 4, asks what do you think moved through the membrane? So hopefully you're thinking water, because water is at a very high concentration outside of this dialysis tubing, whereas it's less inside. So water is naturally going to move from high concentration to low concentration in the bag. And we saw that. We saw a weight gain. Now the second part of activity 4, so this begins question 5, is to take 3 test tubes and fill it with the water outside of this dialysis tubing. And we're going to test to see if starch is there, if iodine there, or if glucose is there. So here is an example of one of those group's tests. So with these, go ahead and complete the questions on page 71. So 6, 7, and 8. This also marks the end of this video. So once you answer those questions, go ahead and start part 2 of the lab.